Good morning, everyone, and welcome to the WRI Research Seminars series. This morning I welcome Simon Michelmore, who's doing his PhD with Matthew Tucker's group, and also he's an employee of SARDI. And today he'll be talking about teenage mutant pulse crops.
Thank you. Yes, I'm talking about teenage mutant pulse crops. So hopefully people understand the reference to Teenage Mutant Ninja Turtles. If not, I'm going to look silly. But yes, this is my previous work at SARDI as well as my current PhD work, both of which are funded by the GRDC and hosted by SARDI and the University of Adelaide. Pulse crops. There we go. Pulse legumes. In Australia, the major ones are lentil, chickpea, fava bean, and field pea. So Australia is the second largest exporter of pulses in the world. We produce 2.7 million tonnes per year, which has a total value of $1.7 billion. And pulse crops have benefits to sustainable farming systems of nitrogen fixation, pest and disease break, and grass weed management. An important part of the crop rotation. Unfortunately, weed control remains a major constraint to pulse production in Australia because they are susceptible to most of the commonly used herbicides, as well as residues of herbicides in the soil from previous seasons, which really limits the flexibility of the crop rotation system. And weed pressure reduces grain yield and quality and can result in complete yield losses if you can't harvest your crop for the weeds. So the photo on the right there is a lentil crop that I drove past earlier this year at Michelmore, and you can see that it is full of wild mustard and prickly lettuce and sow thistles. So herbicides are one of the most effective ways to manage weeds on a broadacre scale. And so about 10 years ago now, a Group B herbicide tolerant lentil was developed, and about a kilometre down the road from this paddock was another one that was a herbicide tolerant variety. See, there's no weeds at all. So these novel herbicide tolerant traits offer new weed control options that can assist in the adoption of pulse crops into farming system. So that herbicide-tolerant lentil was originally released in 2012, tolerant to Group B herbicides, which are acetohydroxy acid synthase inhibitors. And as of last year, 75% of lentils grown in Australia had that herbicide-tolerant trait. Subsequently, in 2019, the fava bean program released PBA Bendoc, which was a fava bean variety resistant to those same herbicides. And that's seen a similar rapid uptake by growers. And it's a really nice complement to all of the other Group B tolerant crops that are available in Australia in other species. So farmers don't have to worry about flightback restrictions from herbicides used in previous seasons or anything like that. But unfortunately, Group B herbicides have one of the highest incidences of weed resistance in the world. So that graph there is showing different herbicide modes of action and the number of weed species in the world that have developed resistance to them. You see, Group B is far above all of the others. So Group B is just one mode of action of herbicides, and there are others that target different biological systems. You've got things like the EPSPS inhibitors like glyphosate and photosynthetic inhibitors like atrazine, as well as the synthetic auxins, which all seem to have a lower incidence of weed resistance. So the Canadians figured out quite a long time ago that all of these Group B herbicide tolerant crops were causing an increase in herbicide tolerant weeds in their system. And that was found to be due to an over-reliance on a single mode of action. And sure enough, we're starting to see the same thing here in Australia, with all these Group B herbicide crops being grown, farmers are more and more relying on that one chemistry to control broadleaf weeds, and seeing a huge increase in resistance in the weed populations. So, to minimise the incidence of those herbicide-tolerant weeds, we need other herbicide options. Unfortunately, there's been no new herbicide modes of action released or registered in the last 30 years. And so we have to take a lot of care to make the most of the herbicides that we have available to us now. So I'm sure everybody's heard of antibiotic-resistant bacteria through overuse of antibiotics. It's the same thing, but with weeds. So to maximise the longevity of those herbicides that we do have, we need to have well-planned herbicide rotations, so using different modes of action season to season, so that we're not putting the same pressure on the weeds every year. We can optimise the rate of herbicide application and mixtures of different modes of action applied together to make sure that we're killing the weeds effectively and not letting those ones with a low level of tolerance slip through. And we can optimise the timing of application, so some of those resistant weeds might be less resistant if you spray them early enough in the season at the right growth stage. We can monitor herbicide resistance so that we can better plan out integrated weed management practices. And there's a service on campus here that Peter Gonzalez runs that provides that service for industry. And we've also included non-herbicide options to control weeds. So things like the weed seed terminator or tilling, which I try to avoid. But an even better way to manage that is to develop crops with tolerances to other herbicides so that we can rotate those herbicides a bit more easily. So these pulse crops are not resistant to a lot of herbicides, so there's an opportunity there to develop novel herbicide tolerance traits to give new opportunities for old chemistries to be used in new ways. So this is where the Mutant Ninja Turtles come into it. So we know from the literature, being comic books and movies and TV shows, that if you put the ooze on some baby turtles, you get superpowers. And so similarly, if we take chickpea seeds and we put EMS, which is a chemical mutagen, on them, and then multiply those chickpeas and spray them in a very large scale in the field with herbicides, we can get different sorts of superpowers, get novel herbicide tolerance traits. So that photo there is a wild type and a mutant chickpea sprayed with synthetic auxin herbicide. See the wild type's killed and the mutant is not. Now we have mutant pulse crops. So this was done as part of a project led by Sally and the University of Adelaide. That started way back in 2009 and it's been going for over 10 years now. That was funded by the GRDC, and that developed these novel herbicide tolerance traits in all those 4 pulse crops I mentioned earlier to a range of different herbicides. So I'm focusing on these synthetic auxin herbicides for this talk. So synthetic auxin herbicides, as the name suggests, are synthetic chemicals that mimic the plant hormone auxin. They come along and bind to an auxin receptor, which results in an accumulation of ethylene and abscisic acid, tissue swelling, repression of photosynthesis, senescence, and eventually plant death. So in a mutant pulse crop, we have target site tolerance where that auxin receptor is either modified or completely knocked out. And that prevents those herbicide symptoms from occurring and the plant survives. But being an auxin receptor and being required for perception of plant hormone, as this AFP45 auxin receptor normally recognises auxin, and that has the normal healthy auxin response, regulates the transcription of several genes that are involved in a range of different plant development processes, including shoot branching and embryo and seed development. So if we modify that auxin receptor to get a herbicide tolerance, what might we be doing to all of these normal auxin response processes? And how do those affect agronomic traits like fertility and plant growth and phenology, and ultimately plant yield or grain yield? And if it is affecting these things, are those effects variable between different alleles of the gene? Are those changes manageable, and are they acceptable to industry? So a small yield penalty is acceptable if you have the trade-off of herbicide tolerance and weed management. So understanding all of this in more detail will help growers and breeders to make a decision of whether or not the herbicide tolerance is worth the trade-offs in fitness and yield. My PhD project is asking a lot of questions around that. And so in field P, I've identified 14 different alleles of this auxin receptor protein. So they are single amino acid substitutions, as well as premature stop codons, and one alternate start codon allele. And they have a range of different effects on herbicide tolerance as well as other traits. So, like any teenager, these plants have raging hormones that are affecting their growth and their development and their behaviour, and we're starting to get worried about their obsession with martial arts. So PBA Wharton was the wild type variety that was mutated, and the mutants have these complicated names. This is abbreviation of 17PAHCL065, which is a bit of a mouthful. So we're going to rename these ones to Michelangelo, Donatello, Leonardo, and Raphael. So you can see there's a range of differences there in their plant type, and this is just 4 of these 14 different alleles that I'll be focusing on. So we want to know about these teenage mutant pulse crops. What are their superpowers? They've got these mutant superpowers of herbicide tolerance. So in a seedling dose response experiment where plants were sprayed with increasing rates of the synthetic auxin herbicide clopyralid, you can see the wild type is completely knackered at 6,400 grams per hectare of the herbicide. And you can see quite severe symptoms even at the lower rates. And in this herbicide tolerant mutant, we see virtually no herbicide symptoms at all, even at rates up to 200 kilograms per hectare. So very highly tolerant. So in this experiment, we're able to calculate the herbicide tolerance level of each allele, which is measured as the GR50, which is the herbicide rate that's required to reduce plant growth by 50%. So we see a range of different herbicide tolerance levels here. This is fold tolerance relative to wild type. See everything from around 6-fold up to more than 1,000-fold for this one that was undamaged even at the highest rate. So here's our 4 favourite alleles here. And we can see that Michelangelo's up here untouched, and Raphael's right down here with the lowest level of tolerance, and we've got Leonardo and Donatello in the middle here. So pretty much all of the alleles have a decent level of herbicide tolerance, but they seem to have varying effects on their branching and other traits. So these teenagers have growing pains. Their branching growth is dysregulated, and that's not too surprising given that auxin is a key regulator of apical dominance, and AFE45 has been previously described as mediating a feedback regulation of the branching hormones strigolactone and cytokinin. But the precise roles of AFE45 in this feedback loop are still a bit unclear. But previous work in peas on this pathway was done with 2 different alleles, and now I've got 14 new alleles, including some nulls that seem to have an even more severe phenotype. And so an opportunity has now arisen to revisit some of that older research with new genetics and new technology to understand that pathway a bit more clearly. So here's the range of different branching severities we see in these 10 alleles here. So this is the branching ratio, which is the ratio of the total length of the lateral branches to the height of the plant. And you can see a range of variable phenotypes here, all the way up to that null allele there that's very dwarfed and branched. But some of these ones in the middle here seem to have quite normal branching, even though they've got a high level of herbicide tolerance. So they might be more acceptable to industry. And we've got our turtles here. So it's looking like Leonardo and Donatello are the favourites here that have got high level of herbicide tolerance we saw in the previous slide, and only a moderate effect on branching. So the other thing we need to worry about with teenagers is birds and the bees. So auxin's involved in reproduction, so it's involved in pollen and ovule development as well as embryo patterning and development. And modifying auxin perception might have an effect on fitness and yield through interrupting this reproduction. And sure enough, some of the genotypes when I've grown them in controlled environments seem to have reduced seed set. So we have a wild type pod here full of seeds, and this mutant that's only got 3 out of 6 or 7 positions with grain filling in it. And this one's only got 1. So clearly some of these alleles do have a significant fitness penalty, and understanding that and trying to mitigate that will be important for actually delivering these traits to growers and maintaining profitable yields as well as herbicide tolerance. So when it comes to yield, there seems to be an effect of these different alleles on both seed size and yield. So this is shown both of these as the percentage of the wild type. So some of them seem to have significantly reduced seed size and yield, and others seem to be yielding on par with wild type. But you will notice that there are only error bars on the wild type here, and that's because this was from some very limited field data that was not replicated. So I'm actually waiting for a rainy day to harvest an experiment in the growth room now to get some better data on yield. But again, you can see all 4 of our Ninja Turtles here are performing reasonably well, certainly not as bad as some of the others. And yeah, so dissecting this yield penalty, looking at the allele frequency in segregating F2 populations, some of these alleles seem to have reduced allele transmissibility. So fewer F2 plants had the tolerant allele compared to the wild type than we would expect for a normal single gene trait. And I'm still gathering data for the other alleles, but certainly Leonardo here has got no problem with allele transmissibility, but some of the others seem to have significantly reduced allele frequency in that F2 generation. So it's been concerning about whether that will be affecting yield in the field. So what to do next, parenting these teenage peas? Well, my PhD is arranged into 4 main research questions. So I want to understand which of these alleles gives the best herbicide tolerance levels. What's the specific role of that auxin receptor in branching regulation, as well as in reproduction and development of reproductive tissues? And ultimately, what is the yield trade-off of herbicide tolerance, and how does that feed into that cost-benefit analysis? So understanding all of these things will help us to identify our favourite character, which will should the breeders focus on delivering to growers. Understanding the mechanisms underlying the fitness costs associated with the trait might help to manage and minimise that fitness cost so that our favourite character can be even further enhanced. And ultimately, we want to give growers the best tools to manage weeds sustainably and to increase the adoption of pulse crops into sustainable crop rotations and prolong the longevity of the herbicides that we have available to us. Thank you to my supervisors, Tim, Matt and Phil, as well as my colleagues at SARDI and the whole Brewer Lab, and everybody for listening.
Thank you, Simon. If you have any questions, can you type them up in the Q&A box, please? I might start just with my limited knowledge of peas. So is more branching actually detrimental to peas, and does that influence the yield?
Yeah, so that's something I do want to try to understand a bit better from my project, but certainly peas have been bred for field conditions to have a certain number of basal branches and aerial branches that seem to give the optimum plant architecture, which is not just about yield, but is also about the crop standability, like getting to stand upright, and there's also implications for disease management. But yeah, obviously more branches might mean more yield potential because we have more flowers.
Yeah.
But whether or not that yield potential can actually be realised with all of the plant's resources being divided into so many branches and so much more leaf biomass.
And is disease more of a problem if it's more branched? Yes.
I believe so, yeah.
Okay. So that's a trade-off again.
Yeah.
Do we have any questions? Okay, question from Ian. So presumably you can offset the yield penalty of the alleles by having heterozygosity as a receptor.
Yeah, so previously in the literature, the branching trait has been reported to be recessive, and so a heterozygous plant should have reasonably wild type growth and architecture and presumably have, a lower yield penalty. I've actually seen in some of my alleles that the trait is sort of semi-dominant or co-dominant. I've seen intermediate phenotype in the heterozygotes. In terms of having a heterozygous crop in peas, I don't think there's much in the way of hybrid pea technology out there, like we have with some other crops like canola. But certainly, if we were able to have a GM crop, we could potentially put in a wild type copy to partially restore the phenotype. But again, the heterozygotes are going to be a lot more susceptible to the herbicide than the homozygotes, which, may or may not be worth the benefit we get in yield penalty.
Do we have any more questions from our online audience? Yeah. Okay, if we don't, I'd just like to thank Simon for his,
Brendan.
Okay, we do have one more question from Brendan.
Yes.
And any early info on differences for phenology or maturity?
Yes, that's a great question, because obviously hormones are involved in a lot of phenology traits, so flowering time and plant maturity, and certainly, yes, I have seen that both in the glasshouse and in the field, that some of these mutants have significantly delayed maturity. So even when the wild type's completely dried off and ready to harvest, we see some of the herbicide tolerant lines are still green and trying to flower. And that's probably due to downstream effects on the regulation of other hormones like abscisic acid. So that is something that's worth taking into consideration when choosing a line for commercialisation and also just managing these as a variety.
And a question from Victor. The traits of interest, branching, seed number are density dependent. It would be interesting to check mutants in a range of densities. It may partially compensate lower yield branching.
So that's a great point, Victor, and it's something that I've been thinking about ever since Victor's talk last week at the Saudi Research Forum. Branching regulation and seed number can be affected by nutrient availability in the soil and competition, both root and shoot competition and light availability. So that's something I've been interested in looking at, is whether sowing density could affect this branching trait. So either a higher soil density would reducing the branching, or a lower sowing density allowing those branches to spread out and have access to more resources to actually realise their potential. I think that's sort of outside the scope of the PhD project at this point, but if Victor's willing to sell a trial for me and help out with that, I'd love to look into it.
Thank you, Victor. So thank you very much, Simon, for a really interesting presentation, and all the best for the rest of your PhD.
Thank you.
And thank you everyone for attending.